This is a demonstration of a piece of software for taking a bunch of papers off of PubMed and turning them into a knowledge graph. Let's look at the pretty pictures first. Let's look at the entire graph, or most of it anyway. Here's the entire graph. Let's zoom out a little. We don't have all the connections between the different concepts. So what I'm calling a concept can be a disease, or it can be a protein, it can be a gene. There's another disease. Some of these relationships between these things are, we don't yet know what they are. The system isn't that sophisticated. It's a work in progress, so it just says associated with. When we click on associated with, there's the relationship. We're very confident the association. Here's the source document, which is a paid paper on PubMed. And this talks about, so let's look and see. All right, this is gastric cancer. So here we are looking at that, and I don't have enough medical background to make a lot of sense of this, but clearly it's related. We can zoom in, we can say, okay, I'd like to look at this subgraph here, so we're going to go in here, get rid of that. If we double click on this, I think. Yep, okay, reset the view. Now we just have that piece of the subgraph. And we can look at immunotherapy it's really a treatment, but that calls it a drug. That's, I guess, a bug. I suppose that's a disease. Pheroptosis sounds like a disease. Immune cells, protein. Well, they're more a cell structure, I guess. But anyway, but we can click on these guys and we get information. And again, we have these things. Confidence in this is a little weak. Usage count tells us, so this was done with about 100 papers off of PubMed, and so this concept only came up once in those 100 papers, so that's the usage count. If we go up to, okay, let's get rid of this. If we go up here and we say, hello, hang on. All right, come on. Let's do it like this, okay. If I ask for cancer, I get these different things relating to cancer, and if we choose one that is likely to, yeah, it doesn't tell you anything about those usage counts, but I think this might, let's look at that one. Yeah, that's not too great. Let's find something more interesting. Well, let's try kidney cancer. Okay, kidney cancer at least. Okay. Let's see if it has a... Oh, it has a usage count of 2. That's good. So that might just mean that it appears mentioned twice in the same paper, since we're only seeing this one paper. Yeah, now that I think of it, when something's very common in the literature, we should see multiple sources for it. Canonical ID, that means that we decided that the MESH website at NIH is fairly authoritative, and it has interesting, useful things to say about kidney cancer. While working on this, I learned that neoplasms is another word for cancer. All right, so, that's that. This is various things to, let's see, if we had more stuff here, we could increase the number of hops and it would grow the graph to show more things. But right now this piece of subgraph only has a couple of hops. Let's do this. Let's load the whole graph, and if we now increase this, let's see if we pick up any more connections that way. This one looks fairly rich over here. Oh, I guess if we're looking at the whole graph, we're not really using the number of hops. So, okay. Okay, let's try this. Let's go to this guy. And if I show the graph with only 2 hops, I'm only going to pick up that, whereas if I now go to 3 hops, it should now pick up the last piece of that subgraph. Okay. And if I go to a lot of hops, it's probably just going to, yeah, that's just an isolated piece of subgraph. It doesn't know about connections between these and other things. Although I imagine mitochondria ought to be pretty well linked to other things. Let's take a quick look at mitochondria. All right, so here's the paper that's mentioning it. Mitochondrial targeting. All righty. All right, go back here. Oops, not that one. This one. This, I'm guessing, that's a drug. This is a procedure, gene therapy, which targets mitochondria, not too surprising. DNA repair deficiencies would be targeted by this gene therapy, or diagnosed by. Sorry. This may be an error. So this is a work in progress. Some of these things are either incorrect or insufficiently specific. Like if we go back to the full graph, you'll see that a great many of these things say associated with, which just means, well, we know it has something to do with it, but we haven't, the ingestion process for pulling in papers hasn't told us that this pathway connects to this disease in a particular fashion. We don't know if it, we do have representations for like increases risk or decreases risk or things like that, but that is not correctly ingested here. What else have we got that's interesting? So we can search by the names of things. Let's see. Yeah, sometimes this thing locks up and I don't know why, so I have to do that. But we can say, what do we know about kidneys? Well, we know there's kidney cancer. Oh, Nova Scotia is a location. I'm curious now. What do we know about Nova Scotia? It must have been mentioned in a paper, but not connected to anything. Yep, nope. But if we click on it, we at least can, oh, here's the paper that mentioned it. Let me get rid of some of these other papers so that we can look at this stuff. And we'll look at this. So here's the paper that mentions Nova Scotia. Yep, there we are. The work was done in Nova Scotia, some of it anyway. And here we have a DBpedia article. So DBpedia is it's part of the Linked Data Initiative, and it's derived from the information on Wikipedia, but it puts it in a form that it's all linked in a machine-readable way. So we have all these things that have to do with Nova Scotia. So that's fun. Let's see, other things that this is doing. If you're a software geek, you may be familiar with the docs view of Fast API. This is all the API endpoints that we're using. We also have a GraphQL view. This lets us look at this lets us look at, oh, okay, that's why that's messed up. Yeah, I made a mistake here. I gotta fix that. But this is basically, so these IDs are taken from authoritative sources like RxNorm and Mesh, and when we can't find an authoritative ID, we give a provisional ID starting with PROV and just a long random string after that. We do accept DBpedia as one of the sources of authoritative IDs. What else do we know here? Okay, that's pretty much it. Okay, great. Thanks very much, and Yeah, so that's now the one interesting thing about this is not, so when you look at this, you're not actually looking at any language model or AI technology. The ingestion process used AI to identify these objects and the connections between them, but at this point, once we've rendered it into a graph, a language model can use that graph, but the graph itself is it's a normal, ordinary database. It's actually Postgres, which is a pretty off-the-shelf typical database. So that's all there is to that. All right, thanks very much, and we're done here.